Hey y'all, it's Ryan, and today I want to talk about one of my papers with some awesome grad school friends, where we evaluated how different strategies that people use to manage their emotions predict nasal cytokine production after being infected with rhinovirus. Cytokines are very important messengers of the immune system that can be either moving freely around in your blood or produced locally in response to a specific injury or infection. So when we say nasal cytokines, we're referring to locally produced cytokines in response to rhinovirus. And we specifically focus on pro-inflammatory cytokines interleukin 6, interleukin 8, and interleukin IL-1 beta here. This question matters because nasal cytokines cause many of the symptoms we experience when we're infected with the common cold. So to give you some background on how we started this paper, we were actually in a statistics class together learning how to model data that has multiple time points and multiple levels. So for example, you might have questions about educational outcomes and look at either the classroom level predictors, so how long a teacher has taught, or the individual student predictors like the child's family socioeconomic status. And that's sort of the traditional example of multi-level modeling, where you have students nested within classrooms. But for biological data, like what we're talking about today, we often have time points nested within people. So, for example, today I'll talk about the level of nasal cytokine production over 5 days while people were quarantined after experimental rhinovirus exposure. This means that we have 200 some odd number of people and 5 data points or days for each person. So we wanted to see if the level of nasal cytokine production was the same or different across time based on what strategies people reported using to manage their emotions. So before telling you more about our project specifically, I'll briefly recap how the original researchers designed the third wave of the Pittsburgh Common Cold Study, which is the data we used for this project. And 213 participants were experimentally exposed to rhinovirus, which is a virus that causes the common cold, and were then assessed to determine who was infected, as well as how severe their illness expression or symptoms were. And there were 3 iterations of the Pittsburgh Common Cold studies, but we're focused here on the third wave. At baseline, people self-reported their age, income, social, psychological, demographic, physical health conditions, and also reported the frequency with which they tend to use different strategies to regulate their emotions. More about that from a special guest in a minute. So then people were exposed to rhinovirus using nasal drops. And after that, they began a 5-day quarantine in a hotel. During the 5-day quarantine, the researchers asked participants to complete daily affect measures. They measured their daily activity and nasal clearance and mucus weight. In our project, we focused on the level of nasal cytokine production each day after rhinovirus exposure.
So what was our question again? We wanted to understand if how often an individual reports using an adaptive strategy to effectively manage their emotions influences local nasal cytokine production during rhinovirus. Emotions are associated with the thoughts and feelings we have every day. And note that this is different from emotion regulation, the ability to effectively manage your emotions. Regulating emotions is vital to health and well-being. So what's an example of effectively managing one's emotions? Cognitive reappraisal is an example of an often adaptive emotion regulation strategy where you change the meaning of the stressful situation in front of you. For example, you could be driving by a car crash and cognitively reappraise the situation by telling yourself, no one was fatally hurt, insurance will help, and everything is going to be okay. Expressive suppression is another strategy that involves inhibiting or hiding an emotion expression behavior and can often be maladaptive depending on the context. For example, if you're upset about something, you may put on a poker face. One way of measuring frequency of cognitive reappraisal and expressive suppression is with a scale or questionnaire, which, surprise, surprise, is called the Emotion Regulation Questionnaire. Once participants fill out this questionnaire, it provides 2 scores. One score indicating the frequency of cognitive reappraisal use, and the higher, the better, and another score indicating the frequency of expressive suppression use. In this study, we used the scale to index our emotion regulation strategy of interest, cognitive reappraisal.
Of the 213 people exposed to rhinovirus, 159 or 75% of them became infected, and 63 or 30% developed clinical cold. That's right, some people are exposed to a virus and don't get sick. And we classified participants as being infected if the researchers recovered the challenge virus from participants' nasal secretions post-challenge, or if participants displayed a 4-fold or greater increase in the virus-specific antibody titer between the pre-viral challenge baseline and 28 days post-challenge. Among those who were infected, some went on to develop a clinical cold. Having a clinical cold meant that their total baseline adjusted mucus weight, so that means summed across all days post-challenge, weighed 10 grams or more, or if their average baseline adjusted nasal mucociliary clearance time was 7 minutes or longer. And we found that people who reported using cognitive reappraisal more frequently actually produced fewer nasal cytokines across the 5 days of infection with rhinovirus, so the common cold. And this was a significant difference overall, both in the clinical cold sample and the infected samples. Once you broke it down into the specific days, you can see that it's really days 3 and 4 where the significant difference in nasal cytokine production occurs based on the frequency with which one uses cognitive reappraisal. Okay, so the reason this is especially interesting is because those are also the same days when people's symptoms are typically the most severe. And remember, it's these nasal cytokines that cause the symptoms that we experience when we're sick with an upper respiratory infection, like the common cold. And unfortunately, our sample size was not big enough to directly test whether nasal cytokine production mediated the relationship between the frequency of cognitive reappraisal and one's symptoms. But this is good preliminary evidence that increased nasal cytokine production that's associated with how we manage our emotions is part of why some people have a much worse experience when sick with the cold than others. And this research is especially cool to me because we're really at the beginning of health psychology and psychoneuroimmunology as a field compared to many others. So yes, this was a sample size too small to test everything that we wanted, and this is only one sample of mostly white, well-educated participants. So it's not the last study of its kind by any means, because we still have so much to learn. But hopefully this gives you an idea of what kinds of questions you can answer and ask around acute infections. So to wrap it up, we found that people who reported using cognitive reappraisal more frequently produced fewer nasal cytokines in the 5 days following experimental rhinovirus exposure, but especially on the days that tend to correspond to one's symptoms. We also replicated past findings that these nasal cytokines are associated with one's self-rated symptom experience during the common cold. All this means that the way we manage our emotions may actually be influencing how sick we get during an upper respiratory illness. All right, y'all, that's all for today. Thanks for stopping by. Please click like or subscribe on this video if you want more videos like this.